everyone, and welcome to ASBMB Journal Club. I am Raj Mukhopadhyay, the science writer for the American Society for Biochemistry and Molecular Biology.
My name is Jeff Hunt. I'm the Society's Public Outreach Coordinator.
So today for ASBMB Journal Club, we'll be discussing biomarkers. Broadly speaking, a biomarker is a molecule. It can be a lipid molecule, it can be a snippet of RNA or a protein, and researchers would like to use these different molecules to track processes that are going on in your body. The processes can be normal physiological ones or they can be disease ones. And basically, researchers are trying to come up with techniques that can accurately track these molecules.
And so speaking of researchers, we have 3 expert panelists who are going to be our discussion leaders today. And so I'm just going to have them introduce themselves one by one. Let's start with Rudy.
Hi, I'm Rudy Ebersold. I'm working at ETH in Zurich, Switzerland, and we're a proteomics laboratory. One aspect of our work is biomarker related for proteins.
Cool. All right, Dan.
I'm Dan Liebler, director of the Jim Ayers Institute for Precancer Detection and Diagnosis at Vanderbilt. We also do a lot of mass spec-based proteomics. We're interested in cancer, particularly understanding how gene expression in cancer translates to cancer phenotypes via proteins and proteomics.
Great. And Karen.
I'm Karen Rodlin. I'm the Chief Scientist for Biomedical Research at Pacific Northwest National Laboratory. I'm really a cancer biologist who uses proteomics, and I'm associated with the mass spec facility here at PNNL, and very interested in the mechanisms of initiation and progression in cancers, particularly ovarian cancer.
Great. So we're going to get started with a couple of questions that we've come up with for the panel, but if you have questions for them, either post them on the Google Plus page, you can tweet us at ASBMB, or you can use the Q&A feature on this Hangout, and we'll try to get all the questions answered as quickly as possible. So if everybody's ready, we'll get started.
Sure. So before we launch into the nitty-gritty here, what would you say are the requirements for a good biomarker? What makes a good biomarker? What are the qualities that you look for? And is there a real-life example you can point to? Who wants to go first?
Sure, I can give you an example in cancer. So a biomarker is some molecule that provides some specific information about either the behavior of a cell or tissue or the likelihood of responding perhaps to a drug. And an excellent example in the cancer field is the so-called ERBB2 receptor, commonly known as HER2, and women whose breast cancers express this receptor at elevated levels at the protein level can be treated successfully with antagonists of the HER2 receptor, such as Herceptin. So that's a very straightforward example of a biomarker. And many people tend to think of blood circulating molecules as biomarkers, but biomarkers can be in the blood or they can be in tissues.
So Dan made a very excellent point describing a biomarker that's mechanistically related to the question at hand. And the question at hand is, what is the best therapy for a woman with breast cancer? And if they have the HER2 receptor, then you know that treating them with Herceptin is likely to be successful, and treating them with an estrogen receptor inhibitor is not likely to be successful. There are other biomarkers that are in the cancer field, particularly in early diagnosis, and we're looking for the presence of malignant tissue. And we're looking for the presence of malignant tissue early on before there may be symptoms to go with the disease. And in these cases, sometimes the biomarkers are associative and really not at all related to the mechanism of the disease. Again, in the cancer field, these would be CA125 for gynecological cancers, prostate-specific antigen for prostate cancer. They have some advantages in guiding diagnosis on the part of a skilled clinician. They also have the problem, the liability, that they are not specific to cancer. And therefore, one of the things that we're always looking for in a good biomarker is something with increased specificity so that the number of false positives is reduced.
Rudy, do you have anything to add to that?
Well, maybe a comment on the question you asked, specifically the requirements for a good biomarker. I think we heard now from Dan and Karen examples of different types of biomarkers or biomarkers for different purposes. And I think they also have different requirements. I mean, the type of markers that Karen talks about, which would be early diagnostic type biomarkers, of course, they would have to be easily accessible in that particular person. You cannot go and do for very early detection. If you basically would like to screen a population, you could not go and do, for instance, tissue biopsies. Whereas in the case where that Dan described, where you might target on an already diagnosed cancer a specific therapy, then of course you would tolerate a more invasive procedure. So the requirements, I think there's no generic requirements. The requirements would matter or depend on the specific application. But for early detection, the one requirement would be that these molecules are detected non-invasively, and that's why so many scientists are trying to go after blood biomarkers or biomarkers in other easily accessible body fluids, which is conceptually simple but technically very difficult. Maybe we come back to this in a few minutes.
Yeah, so I mean, on that point, in terms of the actual biomarkers themselves, how are they initially identified? We note that all of you are using omics tools. Is that really the main way presently and sort of going forward to identify these markers as ways to screen for different diseases?
Well, it's certainly one of the main ways that people have gone after this problem. You can come up with what we would call candidate biomarkers. These would be molecules that you suspect might be biomarkers. You can either discover them de novo by for example, analyzing the target tissue. If it's in the case of a cancer, you might actually try and identify characteristic proteins or even unusual, unique molecules that are produced by a cancer you'd like to detect, and then see if you can detect them in the blood. And this, of course, requires a really good assay. But you could, you could do proteomics to start, or you could do genomics to start transcription profiles. But then you make the assumption that a differentially expressed transcript might be present at the protein level and might be a good candidate molecule. A lot of some of the better biomarkers actually come from mechanistically driven studies of the biology of the disease. You can literally discover these things from the literature. And I think one of the real challenges is the next step, having a good enough assay to be able to reliably and sensitively detect the marker in the tissue. And I think we'll probably end up talking a lot about assays and technology here.
There's another major distinction in discovery strategies, and one discovery strategy looks for a marker, a single marker, and it is often knowledge informed. So this would be looking for the estrogen receptor, the progesterone receptor, the HER2 receptor. It has become increasingly evident from the success rate of existing biomarkers that finding a single molecule that has both the specificity and the sensitivity that you need for screening populations is highly unlikely. So a novel approach to looking for biomarkers is kind of machine learning based or statistically based, trying to put together panels of multiple genes and gene products that may be looking at different processes at the same time to come up with a signature of cancer or a signature of prognosis. And those types of approaches really do require a broad global omics-based discovery strategy, because you don't know a priori what is going to be the most informative set of molecules.
Rudy?
Yes, when we look historically for the molecules or the markers that are being used, especially protein biomarkers that are used in the clinic, they were not discovered by omics approaches. They were discovered in various ways, and to some extent maybe serendipitously. So that's of course not an argument that one should not use omics techniques or omics approaches. But there has been already over quite a few years now, first in the transcript field and now more recently also proteomics, there have been quite substantial efforts expanded to use omics technologies to find biomarkers, or at least biomarker candidates. And I think if we look at the history of the proteomic approaches, particularly in blood plasma, it has been really tough going. I think everyone who moved into that field, including us, initially vastly underestimated the difficulties of finding specific biomarkers, especially for early detection, in the vast complexity of the plasma, which of course also is variable from person to person and is variable maybe even during the day, depending on the state of the person. So these comparisons between cohorts of patient plasma and healthy or control group plasma are very, very difficult, and I think these, if they actually work, they would only work in laboratories which have really highly specialized, highly fast and deep analytical tools. So I would recommend to everyone who is out there, if they not really dedicated to spend an enormous amount of effort to maybe not do a cohort like patient versus healthy cohort study on plasma samples, because the yield is likely to be quite low. Again, I'm not saying this is not a valid approach. I'm just saying it's an approach that takes an enormous effort. Some of the labs that do this can probably attest to that.
So what Rudy said does bring us to a question that we had, is that, looking for biomarkers is nothing new. This has been going on for at least a decade or so. So what are the using proteomics tools is what I mean. So what are the lessons learned from what's happened so far, and how do researchers now intend to move forward, having learned from mistakes?
So one big lesson that I think has come from, in particular, attempts to discover biomarkers for cancer, for example, by analyzing plasma or serum from cancer patients and controls. And the logic at first seemed very compelling, that if we have good technology, we can identify the molecules that are different in a number of people that are cancers, a number of people that are controls, and that gives us our list, and we can sort through those and come up with the winners that become our diagnostic test. And there are technology problems with doing that. It's the complexity of the serum and plasma, as Rudy mentioned. But there's another thing that I think is an overriding negative that's very hard to correct, which is the influence of bias. If you think that your group of controls differs from your group of cancer patients only by the disease, you may be mistaken. There might be other characteristics that influence the differences between those groups. The controls might be people who are not fasted, and the cancer patients might have had their blood drawn right on the morning before surgery. They're fasted, they do have a disease, and they're also frightened and stressed about their surgery. So those are things that you might not think about that create true biological differences between those blood samples that you analyze, and can identify things that are not really related to the disease and the question that you're studying, and send you off down a pathway that's ultimately going to be unproductive. And there are many examples of how bias can creep into experimental designs like this.
So Dan makes an excellent point about experimental design, which is absolutely crucial for proteomic discovery experiments, and I think we've learned a lot about that. But even if you have an optimally designed study, there is the problem of human heterogeneity, which is, I think, what Rudy was referring to as well. So let's pretend that you do have a perfectly designed experiment where each one of your patients who donates blood has donated blood before they've had a disease diagnosis, like the PLCO study, where blood was taken at semi-annual intervals for 10 years. This biggest failure still has been in validating biomarkers that are discovered on one patient cohort in another patient cohort. So if you discover your biomarkers in Switzerland with people of a particular genetic and ethnic background, there may be their response to insult or injury or inflammation may be different than a separate population with a different ethnic and genetic background. So that if you try to take a biomarker set that was discovered in Texas and validate it in New York or in Seattle, you may get differing results. And that points to an area of needed technological advance. And that area of needed technological advance is being able to have the throughput to do enough subjects that you can at least include a reasonable sampling of human heterogeneity in your discovery set.
Yes, so I can maybe also add something to this question from Raj about lessons learned. What I think a couple of lessons that we learned include the following. So we learned that we need to have a very clear-cut question. This goes again to experimental design. And I think we learned also from genomics that what appears to the pathologist as a particular type of cancer, or maybe even at a particular stage of cancer, at the molecular level may be not homogeneous and may be different types of classes. And of course, if we cannot ask the specific question, we think that we just can compare proteomic profiles with all this variability that was just discussed, then I think it is very likely that we're misled. So one of the lessons we learned, and certainly that we incorporated in our work, is to basically abandon the search of patterns in cohorts of case and control and to go towards mechanistic analysis and basically do a hypothesis-driven approach in plasma samples, where we would have a list of candidates that may come from the literature, that may come from genomics, that may come from tissue proteomics, or a combination thereof, and then go in sufficiently sized cohorts to find out whether these patterns really, whether these particular proteins that we have postulated to be candidates really go or correlate with the disease. So this has been certainly in our hands more successful, more promising. It is still a technically difficult thing to do. We use for that kind of hypothesis-driven approach different mass spectrometric techniques. Maybe we come to that, but just briefly, if one does the cohort comparisons, like cases and controls, one would likely use some form of quantitative discovery proteomic technique. And there is many varieties of those, but they basically try to cast as wide a net as possible into the corresponding or the proteome that's being studied. Whereas if we are using a hypothesis-driven approach where we have a list of candidates that we want to consistently quantify across a large number of cohort individuals, healthy and controls, then we would more likely use different type of mass spectrometry, targeted mass spectrometry, which specifically goes and quantifies only those proteins on our list across the samples, but not any of the other proteins that also will be in there. So the research strategy will also determine the techniques that are probably most optimally used or suited to answer the question.
Great. So before the next question, I just want to remind anybody watching that if you have questions for our panelists, you can either post them on our Google Plus page or within the Hangout Q&A feature. So sort of following up on our last question, how are biomarker-driven approaches to diagnosis of disease compatible with symptom-based diagnoses? Where do they differ? Where do they overlap? And how can they be used synergistically, not just by researchers, but also by the doctors who are actually in the clinic treating patients?
I think there's an excellent strategy here and an excellent analogy. Symptom-based diagnostics require the skill and the expertise of a very experienced clinician who can sort through multiple layers of information and weight them differently, and who looks at a combination of things. There might be different, we could call these biomarkers. One's temperature, one's white cell and red cell count, whether one's been gaining or losing weight or has a stable weight, whether one's fatigued or not, blood pressure, heart rate. These are all biomarkers that we've been using for centuries and that internists and oncologists use as for symptom-based diagnosis. One of the problems with cancer is that the most lethal cancers, pancreatic, ovarian, sometimes colon, can be silent until very late in the development of the disease, at which time it's very difficult to cure them. They cannot be completely removed by resection. They've metastasized. And so that's been one of the real drivers for developing biomarker science, is to detect cancers at earlier stages where treatment is less drastic and more likely to succeed. But I think even using a hypothesis-driven approach, as Rudy described, we need to start thinking like a good internist and not rely on one hypothesis, one signaling pathway, one mechanism involved with cancer, but start looking at the fact that there may be multiple functions that are disarrayed: proliferation, invasion, apoptosis, loss of differentiation, and begin to compile panels of biomarkers that are not random patterns of features, but that represent the most robust signature of a particular process. And we put these processes together in a way that completes our picture of cancer as a holistic disease.
You know, I think it's a really great question about how would we integrate, you know, biomarker tests, let's say new biomarker tests coming out of all these new technologies. How would we integrate those with the basic features of clinical practice and diagnosis? And I think that that really highlights where the opportunities are, in my view. You know, and when I got into this field, I certainly had this concept in mind of, you know, getting your car license renewed and giving a blood sample and finding out if you've got cancer. But maybe the more realistic scenario is thinking of biomarkers as adjuncts to clinical decision making. And this actually allows you to identify, as Rudy pointed out, the very clear, pertinent question, and then have the work on a biomarker really be driven by the need, the clinical need. And in many cases, you know, you've got other data available to the clinician, whether it's, you know, physical assessment or other laboratory data, that puts your biomarker measurement in enough context to remove many of the ambiguities that would that would really confound interpretation of your test when there's no other information.
Okay, so it looks like we have a question from the audience. It's open to all three of you. What is the biggest bottleneck in acceleration of a therapeutic from identification to a clinical application? Is it the engineering of the targeting therapies or the identifying the biomarkers? What's the biggest bottleneck here?
Well,
I went to a biomarker journal club and a therapeutic journal club broke out. Like the old joke about the hockey game, right?
Yeah.
Went to a hockey game and a fight broke out. Now, great question, because I think one of the real applications for biomarker studies is to identify the right patients for a trial of novel therapeutics. I think one of the bottlenecks in therapy has been, as therapies become more and more targeted, not everyone is going to respond to that particular therapy. So identifying the appropriate population for that therapy is very important. And my favorite example here is Gleevec, which is one of the first targeted therapies in cancer that's really worked. And if Gleevec had been subjected to a clinical trial in all lymphomas and leukemias, it would have failed the clinical trial. But we had a molecular marker for chronic myelogenous leukemia. We had the Philadelphia chromosome, and it had a particular molecular defect, so you could target that defect and you knew whom to treat.
Any other?
Maybe you can also add a couple of statements here. This is actually, I think, a really interesting question, because I think the field is moving away from non-discriminate screening into this mechanism or pathway or process-based analysis. And of course, it is exactly those pathways or processes that are affected by disease that we would expect to leave a footprint, which are at the same time also targets. And especially, I think that the strength of the proteomic approach is that the molecules that may be biomarkers may indicate the range or distorted or dysregulated pathway at the same time may also be biomarkers and targets. So the two, I think, are drug targeting and biomarker measurements are maybe converging biologically onto the same subsystems of the cell.
I think somebody's a little close to their mic. I don't know who it is, but we're getting some ruffling.
Okay, hope it's not me. Then then I also have a short comment to the question just before about symptoms. I think we cannot be under the illusion that molecular markers which come out that would show up like at the airport when you go through screening and they swipe your bag, that it will just say red or green and you either go home or you don't go home. So this is clearly already borne out with the markers which are used in the clinic. They are not red or green signs, they are maybe warning signs or hints. And the one, I think, very celebrated case is PSA, which is at some level a very good marker because it captures most of the individuals that actually do have prostate cancer. But it also captures a lot of men who do not. And that led to then an interesting discussion about costs, whether the test actually should be deployed, because what would one do with the high number of over-predicted or over-diagnosed individuals? That leads to follow-up examinations, leads to follow-up costs. It has its own hazard to do these follow-up examinations. So it's a very complicated field, but I think in the middle of the whole enterprise is still going to be for a while the physician, not a computer, who, as physician, who needs to synthesize all this information, but who also needs to be educated what actually these tests can do. The idea that one would just throw something into a mass spectrometer or another instrument and it lights up green or red, I think is at this time not on the horizon. I think it's a dangerous proposition. Data should be synthesized, analyzed, and interpreted to the patient by a physician who has a lot of experience and also knows a lot of other symptoms and knows to weigh them.
Great. So we have another question from somebody in the audience. They're asking if the panel can comment on the potential value of metabolomics profiling in biomarker identification. Sort of wondering, in the future, will it be essential to combine metabolomics, proteomics, genomic data in order to identify these markers to sort of help provide some clinical insight?
So I think metabolomics, like proteomics or genomics, gives you another look at the biology. So from a different perspective. I mean, it's all reflecting the behavior of a system. And I think that technology for making measurements of metabolites has improved a lot, just like has for proteomics. One of the challenges with proteomics and even some kinds, metabolomics and even some kinds of proteomics, is making sure that the information that's contained in that sample is still accurately reflects the biology at the time the sample was taken. We know that, for example, if in tissues that have been subject to ischemia, if you cut out a piece of tissue or take a biopsy and you don't freeze it right away, phosphorylations on proteins can become somewhat scrambled. And nobody's really done much of this in the case of metabolite profiles, but one could easily imagine that metabolite profiles could become somewhat scrambled by stress responses and other things that happen after you harvest the sample. So in principle, metabolomics, I'd say 2 thumbs up, but there's going to be some practical challenges to make sure that in real diagnostic samples, what you measure actually reflects the biology accurately.
So we have to not forget that biomarker studies are also a form of investigation into the mechanisms of disease, particularly as we get the capability to integrate genomic, proteomic, and metabolomic data. So given the vast number of metabolic derangements in cancer, I think that gathering metabolite samples and analyzing them together with the genomics and the proteomics is going to help us get a more complete picture of the disease. Whether the metabolites themselves become the biomarker that's used in the clinic, alone or in conjunction with proteomics, is an open question right now. And Dan, you've raised some great questions, some great important points that we need to think about in all of the experimental design for our discovery efforts. But I think already we're seeing that when you combine genomic, transcriptomic, and proteomic information in the same analysis pipeline, that we're learning much more about the disease than we did from any one input alone.
Yes, so I think I'm also excited about metabolomics. There's a lot of progress in the field, and I have probably a couple of additional points to mention. It also matters where one is measuring metabolites, because in contrast to proteins and gene transcripts, where you can say from what species they come from, you can't really say that from metabolites. So if someone is, for instance, interested in stool samples and try to find biomarkers in stool samples, which is actually quite common, especially for colorectal cancer studies, then you might have to consider that there is also a whole microbiome world in that particular body. And so you will not necessarily be able to say whether some of the metabolites are from the host or from the source. And so that, of course, depends on what the tissue or what the sample is, but one needs to recognize that you cannot necessarily say what species or what sample the metabolite is coming from. Then I would like to reinforce the notion that was just mentioned also by Dan and by Karen, that the metabolites are of course not just appearing out of the blue, but they are the consequences of enzyme action. In many cases, this enzyme action is actually known. One knows what enzyme or enzymes can produce a particular metabolite, so one can use the metabolite or view the metabolite essentially as a readout, a functional readout of some pathway or enzymatic system. And we actually just did such a study where which is related to diabetes, where we could clearly show that a genetic variant in a mouse cohort dysregulated through genetic variation, and that was then detectable by the product of these enzymes. Now you might say, interesting, in mouse, we're not interested in mice, in mouse disease, but interestingly enough, exactly the same metabolite was then also found to be associated with human pre-diabetic condition. So clearly this was a case where a connection could be made, a functional mechanistic connection between genotypic variation, where one allele introduces or generates a higher level of a particular enzyme, which is then detectable by a higher level of the product of this enzyme, the metabolite. And I think this is a nice example, and we will see more of those. And it's reassuring to see that all these levels of expression and activity fit together into a particular readout. And that's why I think these metabolomic studies are so exciting, because they provide another angle, another window into the same room.
So before I ask my next question, I just remind people that they can post their questions to the Google Plus page or tweet them to us at ASBMB, and we will ask our panelists to answer the questions. So one question, I mean, we've mentioned, mass spec has been mentioned on and off throughout this entire discussion. So can you describe what the state of the art is now in mass spec and why it is one of the important technologies in this kind of studies?
Well, maybe I can just give a little overview. Basically, there are sort of two broad mass spec platforms that are used in the case, at least, of protein biomarker research. And one is sort of a global discovery platform, and this is usually used to interrogate cells or tissues or biofluids for their content. It's commonly known as shotgun proteomics or global proteomics, and Rudy mentioned it before. There are several ways to do that, but what it does is it gives you a broad survey, and I think the state of the art for sort of average samples that are biologically or tissue cell relevant you can, for a single sample, you can easily identify, 6,000 proteins or more, and you can get quantitative comparisons, or at least approximate quantitative comparisons or estimates between sets of samples. And this is kind of conceptually analogous to a microarray experiment in transcript profiling. And then the flip side of this, the complementary technique, is targeted analysis, where you measure individual peptides that are representative of their corresponding proteins. And this is known as SRM, or selected reaction monitoring. There are a couple other variations depending on the kind of instrument you're using. But this is sort of conceptually analogous to the RT-PCR for a targeted measurement of a particular mRNA transcript. And these measurements aren't able to detect as many things, but they're much more precise. They can be made much more high throughput. And I think these are the measurements that will ultimately replace the Western blot and immunohistochemistry and many of the other sort of antibody-based protein measurement techniques. And one other thing I could say about these is that they're very important in biomarker development, because you can develop a reliable assay using these targeted methods, whereas waiting for a good antibody from Mother Nature might be a fruitless long wait.
So along those same lines, let's talk about sort of the regulatory process in terms of detection and approval of these biomarkers in the clinical trials. Has that been a bottleneck, or is it more on sort of the research side of things?
Well, I think the regulatory bottleneck reflects a lot of the research issues. So it's clear that you will never get a biomarker approved by the FDA until it has been validated in multiple independent cohorts. And that reflects the biological truth that you need to do that. And so you have those technical problems associated with human heterogeneity that has made that a hurdle technically and scientifically for the biomarker community. I really don't think the problem is as much on the regulatory side as perhaps a failure of some people on the assay development side to fully understand what criteria they were going to have to meet.
Yeah, so maybe I can add to that as well quickly. I think Karen nicely summarized the regulatory issues. I think there's also some kind of science logistical issues, because I think it turned out, and we learned this also the hard way, that it is very tempting to, let's say, put a student or a postdoc onto a project, a biomarker project. And I think for those who are out there who are students and postdocs, I'm not trying to discourage them from doing this, but I think they need to titrate their expectations. It is simply not a realistic expectation that a student can start in the first year of his PhD study with a biomarker question and see at the other end an FDA-approved test to come out. This, of course, would be very, very likely, very nice, and it would be highly desirable, but it's not likely to happen simply for reasons of time. So I think it is important if one enters such projects, especially if one is a student or postdoc, with expectations how far a project could be driven within the confines of a typical research group. Because if one needs to go to the regulatory steps that Karen talked about, then one would all of a sudden need to recruit multiple cohorts. These cohorts might have to have increasing numbers of participants, maybe hundreds eventually. And so it becomes not really a scientific issue anymore to drive a biomarker validation from the laboratory to the FDA. It is more also, or equally also, logistical issue and a cost issue. And it may be necessary that different structures or different organizations will do subsequent steps. And so my main message is that if one goes into this work as a student or as a postdoc, then one might want to clearly think about how far this could be driven and when it is a good time to maybe hand it off to the next stage, so that the expectations are kind of managed. Then I have a comment also to Dan's, or an addition to Dan's statement, his very nice summary of the mass spectrometric world. I would just like to add that there is a couple of additional or maybe niche techniques, but which are available, which have, I think, quite nice potential. The one is a combination of immunoassays and targeted mass spectrometry. So this is referred to as immuno-MRM or immuno-SRM, and the idea there is simply that one would use a not really highly specific, or not necessarily highly specific antibody to enrich an analyte and then analyze and quantify it in a mass spectrometer. So these are particularly powerful approaches in cases where very low-level potential biomarker proteins need to be quantified across cohorts. These are very nice, these are very powerful techniques, but of course they need, in addition to the development of the MRM assay, they also need the development of an affinity reagent that's suitable for the task. But in cases where really low-level proteins need to be consistently quantified, this is a very promising approach. Then there is another approach emerging, which is referred to as data-independent analysis, or one implementation of that is a technique we call SWOT mass spectrometry, which is basically has a similar performance profile in terms of sensitivity, dynamic range, and quantitative accuracy, like SRM or MRM, except it can do it at a much larger number of analytes. So the performance, the sensitivity is not quite as good at the moment, but that probably will improve. But it is a technique that can basically multiplex a much larger number of proteins than the SRM technique can with similar reproducibility and dynamic range and sensitivity. So this is all relatively new, but it may be a method that kind of has that combines the advantages of the targeted SRM technique, which is with its accurate quantification, the reproducibility, and the advantages of the discovery techniques, which can analyze in a single go very large numbers of proteins. And that's probably something that will develop quite quickly in the next few years.
Okay, well, we're getting some questions flooding in. Do you want to ask?
Yeah, so I guess the one we'll ask, so somebody asked what informatics tools are either optimal or required to integrate all these different omics approaches from biomarker studies to really look at a mechanistic level the behavior of a system. That's, a small question.
Yeah, no, there are so many tools out there, and they're being developed so rapidly. Almost every lab that does this kind of study has one or more very bright young computational biologists who are developing tools. There are some suites of tools out there that one can buy commercially, Metacore, GeneSpring, IPA. I'm sure that Dan and Rudy can come up with a longer list. I would definitely urge the students out there listening that if you want to be marketable, combine a very strong biology background with a strong computational background, know how to write code, know how to write in R, and you probably have your job made.
That's great advice. Dan and Rudy?
I think the search for tools can be daunting if you start out looking for tools. And my suggestion for people who are interested in doing a certain kind of investigation is find the papers that describe something along the lines of what you would like to do, and then look at the tools that they used and what information those provided, and then use that as your starting point for tools. And one other observation I would make is that there is a wide variety of tools that could come from different sources, and I think many of the tools that are most useful and have sort of been the most thoroughly evaluated and tested are tools that come from consortia of investigators and tend to be more often open source rather than commercial product tools. This is certainly true of the big tool suite that accompanies the genomics data from the Cancer Genome Atlas and the National Cancer Institute Clinical Proteomics Tumor Analysis Consortium that Karen and I both lead teams that are working in that consortium are using and actually developing tools that complement the proteomics for doing cancer genomics proteomics integration. And the nice thing about those kinds of tools is that they're being used and evaluated in vetted by a relatively large number of investigators. A great example is the Skyline tool for integrating quantitative mass spec data. This was developed by Mike McCoss's group at University of Washington through the CPTAC program previously, and it's it's open source, it's highly community supported, very responsive to people who do quantitative mass spec, and it's a great example of a success story of informatics.
So maybe just to add a couple of points also here to these great comments, I would say the following. So don't really think about tools, think about pipelines. It's not really useful to download a tool that does one step in a complicated process, even if this is a very well documented tool, because you will inevitably get stuck somewhere down the road, because these analyses that, especially if different types of data will need to be integrated, are all multi-steps, and the steps need to talk to each other. So one should think of pipelines rather than individual tools. Then it is certainly in the mass spectrometry field, and certainly with respect to data integration, so that the open source community is far ahead of what instrument manufacturers can produce, because they are usually focused on making terrific instruments, but for them software development is a huge challenge, especially if it goes further and further away from the actual instrument. So I would of course, if there is good tools that are close to the instrument, let's say from data acquisition and denoising and de-isotoping or whatever these steps are, the instrument manufacturers may have very good tools, but if it comes to further instrument distant questions like data integration, statistical models, I would definitely recommend you would look at open source tools, and what the ones that Dan described for targeting is certainly an excellent choice. Then the third point I would like to make is data integration across levels only makes sense that if the quality and the statistical soundness of the data at each level is known. So in each level, if one compares, for instance, transcript, proteomic, and metabolomic data, and wants to do some kind of correlation, this correlation will be only meaningful if at each level a sensible error model is being determined, so that one understands what is the variability in technical variability, the biological variability at each of these levels, so that one can do meaningful integration. If you don't know the technical variability at each one of these levels, the integration on patterns arising from it may be completely meaningless, because we may compare actually highly quantitatively accurate results on one level with noise at another level. So I think if someone is going into this complicated business of data integration across platforms, across levels, one would need to work, make sure that the data analysis pipelines at each level have a sound statistical base, so that one can know what is the variability, where is the variability coming from, and what level of variability is actually biologically meaningful.
Great. So we have a number of additional questions, but I think we're out of time here. So unfortunately, we won't have them answered live, but we will follow up with the panelists, and we will post the responses on the Google Plus page so that everybody can see them. I just want to thank Rudy and Dan and Karen for taking the time to talk with us today.
And if you are interested in joining us for future ASBMB Journal Clubs, please check out our website, asbmb.org, for future announcements. Thank you very much.
All right, we'll see you all next time. Thanks, everybody.